Good morning everyone. In this video, we will start with a new topic of the chapter Plant Tissue. The name of the topic is Secondary Growth in Dicot Stem. The main objectives of this topic includes types of vascular tissue, secondary growth in dicot stem, and cock cambium. Before starting with the topic, we must know what is the difference between primary and secondary growth. The lengthwise growth of the roots and stems of the plant with the help of apical meristem is called primary growth, whereas the growth in width of plant with the help of vascular cambium is called its secondary growth. The tissue involved in secondary growth are the two lateral meristems, that includes vascular cambium and cock cambium. Vascular cambium. The meristematic layer that is accountable for severing of vascular tissue, that is xylem and phloem, is called vascular cambium. In young stem, it is present in patches as a single layer between the xylem and phloem and forms a complete ring in the late stages, which is called as cambium ring. Vascular cambium is further of two types, intrafascicular cambium and interfascicular cambium. Intrafascicular cambium is the cambium which is primary in origin and it is present in between primary phloem and primary xylem, whereas interfascicular cambium is the true secondary meristem. It originates from parenchymal cells of medullary rays region. It lies in between the vascular bundles. In general, we can say that intrafascicular cambium is the cambium which is present inside the vascular bundle, whereas interfascicular cambium is the cambium which is present in between two different vascular bundles. In this picture, you can clearly differentiate between interfascicular cambium and fascicular or intrafascicular cambium. Here, a strip of meristematic cell that is present between primary phloem and primary xylem of a single vascular bundle is called as fascicular or intrafascicular cambium, whereas a strip of meristematic cell that is present between two different vascular bundles is called as interfascicular cambium. Now, let's see how secondary growth takes place in a dicot stem. As discussed, in vascular bundles of a dicot stem, the cambium is present in between xylem and phloem. It is known as intrafascicular cambium. During secondary growth, few parenchyma cells lying between two adjacent patches of fascicular cambium of vascular bundles become meristematic and produces interfascicular cambium. Both the intrafascicular cambium and interfascicular cambium unite together to form a complete ring called cambium ring. The activity of cambium ring give rise to secondary growth. The cambium ring acts as meristem which divides. The cambium layer consists of a single layer of cells called meristematic cells of cambium ring. These cells divides in direction parallel to that of epidermis. A cambium cell divides into two daughter cells, one of which remains meristematic and other differentiate into secondary vascular tissue. The cells formed towards inner side develops into secondary xylem. Likewise, the cells which are formed towards outer side develops into secondary phloem. Normally, more secondary xylem cells are formed towards the center, due to which cambium ring moves towards the periphery. Due to the formation of secondary xylem and secondary phloem, the primary xylem and primary phloem, which were initially closed, moves towards inner and outer side respectively. As a result, they become separated apart. These layers of secondary tissues are gradually added to the inner and outer side of cambium continuously throughout the life of plant. Hence, a large number of secondary xylem is produced compared to that of secondary phloem. The secondary xylem thus produced soon forms a compact mass, due to continued formation and accumulation of secondary xylem, the primary and secondary phloem gradually gets crushed. But the primary xylem more or less remains intact in and around the center. At some places, a narrow band of parenchyma is formed by the cambium. Actually, vascular cambium consists of two types of cells, fusiform initials and ray initials. Fusiform initials are the narrow, elongated cells that divides to form inner cells and an outer cells. The inner one gives rise to secondary xylem and the outer one to secondary phloem. Whereas ray initials are spherical cells which divides to form parenchymatous cells which give rise to rays in between the neighboring xylem and phloem. These ray initials of the interfascicular cambium, instead of forming secondary xylem and secondary phloem, forms some narrow bands of living parenchymal cells. These forms 2 or 3 layers of thick radial rows that passes through the secondary xylem and the secondary phloem and are called secondary medullary rays. These secondary medullary rays provide the radial conduction of food from the phloem and water and minerals salts from the xylem. The activity of cambium is affected by variations in temperature. In moderate climate, the cambium becomes more active in the spring and forms greater number of vessels with the wider cavities, whereas in winters becomes less active and forms narrower and smaller vessels. The wood formed in the spring is known as spring wood or early wood, and that formed in the dry summer or cold winter is known as autumn wood or late wood. These two kinds of wood appear together as a concentric ring known as the annual ring or growth ring, as seen in transection of the stem. Successive annual rings are formed year after year by the activity of cambium. The growth of the successive years appears in the form of concentric or annual rings, each annual ring representing the 1 year's growth. The age of the plant thus can be approximately determined by counting the number of annual rings. Hardwood can be further differentiated into ring porous wood and diffuse porous wood. In the temperate regions, the hot and cold seasons are very distinct. This forms concentrated vessels in early wood, clearly distinct from that of late wood. Such wood are known as ring porous wood. Whereas in the tropical regions, seasons are not clearly demarcated and hence the vessels are evenly distributed both in early wood as well as the late wood. Such woods are called diffuse porous wood. Formation of heartwood and sapwood. In old trees, the greater part of secondary xylem is dark brown. This happens because of deposition of organic compounds like tannins, resins, oils, gums, aromatic substances, and essential oils in the innermost layer of the stem. Due to the presence of these substances, the wood becomes hard, durable, and resistant to attacks by microorganisms and insects. This region is composed of dead elements with highly lignified wall. The wood in this region is called heartwood. The heartwood does not conduct water but gives mechanical support to the stem. The peripheral region of the secondary xylem is lighter in color. This is known as sapwood. Conduction of water and minerals takes place through sapwood. This picture is depicting a cross section of the wood of old tree. You can clearly see the inner region is quite dark in color. This is called heartwood or duramen. Its color is dark due to the deposition of tannins, resins, gums, etc. The heartwood ceases the function of conducting tissue and simply provides mechanical support to the stem. However, the outer region of secondary wood, which consists of younger living xylem cells, remains light in color. It is also called as sapwood or laburnum. It functions as the conducting tissue and also as food storage tissue. Now, let's see the secondary growth in extrastellar region due to The activity of cork cambium. The marked increase in diameter or thickness of stem brought about by the secondary thickening exerts a great pressure on the outer tissue. This results in the rupture of cortex and epidermis. The outer cortical cells become meristematic and begins to divide. This is known as cork cambium or phellogen. The cork cambium divides to form secondary tissue on both the sides, that is internal and external. But its activity is more on outer side than on inner side. This phellogen is composed of a couple of layers. This is made up of narrow, thin-walled and nearly rectangular cells. The phellogen cuts off cells on both sides. The outer cell differentiate into cork or phellum. The inner cells differentiate into secondary cortex or phelloderm. Cork is impervious to water due to suberin deposition in the cell wall. The cells of secondary cortex are parenchymatous. Phellogen, phellum, and phelloderm are collectively called periderm. Due to the activity of cork cambium, pressure builds upon the remaining layer which are peripheral to the phellogen. These layers finally die and slough off. As already discussed, the cork cambium divides to form secondary tissue on both the sides, internal and external, but its activity is more on the outer side as compared to that of inner side. The cells formed on the outer side constitutes the phellum or cork, and those on the inner side form secondary cortex or phelloderm. The phellogen, phellum and phelloderm collectively forms a single layer which is called as periderm. The bark is the term given to all the tissues outside the vascular cambium, and hence it includes periderm and secondary phloem. The various layers in the bark are dead phloem, a portion of cortex, hypodermis and cells of outer layers. All tissues which lies exterior to the vascular cambium are called bark. Bark formed early in the season is called soft bark, while one formed late in the season is called hard bark. Lenticels. The phellogen cuts off closely arranged parenchymatous cells on the outer side instead of cork cells. This happens at certain regions. These parenchymatous cells soon rupture the epidermis. This leads to the formation of lens-shaped opening called lenticels. Lenticels permits exchange of gases. Lenticels are usually found on the bark of old woody stems. This is all for the secondary growth in a dicot stem. Thank you everyone.